We're quite excited to have the talk by Dr. Rajaprabu N. He is an assistant professor, uh, and he is also the head of the department of the botany at Bharti Vishwavidyalaya, Durg, Chhattisgarh, India. Today he's going to talk about the modern gateway to lichenology. He'll share some of his ideas about how to use chemistry, how to use genomics for in phylogenetic analysis leading to species delimitation, I believe. Thank you, Dr. Rajaprabhu, for agreeing to give this talk. The platform is is handed over to you. Thank you.
Yes, sir. Thanks for your kindful introduction to about myself. And it is a privilege to present towards the current science community. So it is very kindful thanks to you. And at the same time, I want to, it is not at all new things, but there are some myths are there, particularly field of lichenology, because it is like a biological orphans. So in the case, we would like to present in behalf of Micro-Asia. So it is a very good platform to learn new things as well as many myths can be break here. So like a regular pathway. So because of the scientific community, day by day, it's giving some very good, interesting things, especially in the genomics based. So so far there is a myth on the culturing of lichens. At the same time, in the DNA or else the molecular based studies in lichen. So in this case, I am just start about the classical taxonomy of lichenology. So first we go to the field and we collect some of the lichens. In the second picture, I have collected and mentioned only the basic matters like crustose, foliose, and fruticose lichens we know. But in the bottom side, there is another case. So leprose and fibrils types and the squamulose types, which is not aware of many of the mycologists. So I'm sorry to say this because we people are worried about it is a very difficulties to learn and study about the taxonomy of the lichen. So in the secondary case, in the it is very basic things. Comparatively, the other organisms like plants, animals, insects, including the mushrooms as well as here the lichen herbarium will be totally different. This so these are all the steps we followed for the classical taxonomy, anatomical sections, as well as the spot test will be have been to do, and the third thing is crystallization as well as the thin layer chromatographic technique. So then we used these lichens in different applications. So these are the very basic things only, but that is a huge revenue for particularly the tribes. So the native people are using this. At the same time, the folklore also using this case. In this 17th century itself, it was going on by the father of lichenology, Eric Carriers, so the student of Carl Linnaeus. So this is a microbial biota. You people are very known. There is a photosynthetic partner as well as the mycelial partner to be storage the food materials. This thing is, it is happening with sexual and asexual reproduction for the lichenization product procedure. This is the gateway. We have to understand what are the biota, which means biological organisms are living in the thallus. So then it goes for the in 2017, scientists have been identified about virus, which was living but not contaminating and not destroying the thalli. And it was published in the MDPA journal that called the viral, I think so. So in 2017 itself, it gives the curious to study about this thing, and there are huge number of bacterial strains also found out by the different global lichenologists. So, and this is very common thing. We know all people are very aware, very familiar with the algal diversity in the lichens. But the thing is, how many lichenized algal species will be there? Still, it is a myth. So that is going on here. At the same time, my study found with the endologenic fungus, which will be looking as a in the left side is the Permotrima reticulatum, and the right side is the Funduplicans. The both are have been found and diverged with full number of fungal strains, particularly in the Permotrima reticulatum was very slow growing like that, but it has profoundly this this was very profusely founded, and it was growing in very fast pace. At the same time, it more than in the metabolite base, it was very huge. So when the Ramalina conduclensis, it gives them it is appearance like gravidacy group. So one of the very familiar thing, and the thing is, not only the actomycetes case, including the basidial lichen also be found in the global area, but so far in India, only I think still the research was going on. Only few things have been founded, but so far it's not published. At the same time, in this case, here there are some camouflages. It is like pseudolichens, like frog and moths, and many of them are found in the case. And here, lichen takes more than two tango, which means lichens not only two organisms, including bacteria as well as molds also to be found, including the viral strains also have been founded. So at the same time, one of the molecular evidences says so regarding the Pyrenomycetes, one of the oldest plant which has been thousand for nearly almost 1200 years old, which means not 1200, 1200 million years old bacterial strains have been found from the lichenized material. So, and this is one of the oldest fossil, and still it is surviving and is vastly used in the tropical countries as well as the subtropical areas. The name is Lobaria pulmonaria. So it is called the lungwort lichen, and this lichen is almost 400 million years old. So the study was conducted in Scotland, and this also have been going on that. At the same time, one of the scaly lichen are the um, what we call the squamous lichen, the name of Cladonia, which is called the cup lichens. It gives more than 69% only the chlorophyll will be presented, which means the algal coordinates will be more. At the same time, the eukaryotes, like fungal components, will be just 27%. But we people are thinking about these things, only 10% of algal component and 90% will be held by the only fungal components. But here, the myth was destroyed and reproved with this research. And the same thing in India, Dr. Rajesh Kumar from Agartha Research Institute, he have found about published a new species from the Graphidaceae family, and it was collected from the Western Ghats. But because there are 10 lichenogeographical regions in India, particularly Eastern Himalaya and the Western Himalaya. At the same time, the more distribution have been found from in Western Ghats only, because it was one of the oldest Lemurian continent connected area. It's like a myth, but still we people are studying in that case. So here the Permiliaceae as well as the Graphidaceae totally parallel organisms, like in foliose lichen, another one is in the crustose lichen. But here the um, four part roots, which is means that flats have been served the ascomycetal region. The um, ascomycetal tree have been showing details of Lichenology. So at the same time, Lichenomycetes also been founded, but in the publication have been made by them with very minute organism, almost 1 or 2 centimeter will be the whole thallus will be founded. At the same time, this one is about Cladonia, and this Cladonia have been found with almost 8 species, and the maximum number of thing was founded with cilia cut as well as the flagella proteome. In this case, the chlorophyte cilia was found more than 38% comparatively about remaining things. Everywhere it was connected. And this case, the Diorygma Karnatakensis, at the same time, Diorygma Minisporum was founded from the isolation of lichenized fungi. It will be mentioned in country of India. At the same time, there are many other regions also, like South Africa, Kenya, Brazil, Argentina, and especially Thailand. So here, Robert Looking have been initiated the things, I think, and he is one of the pioneer person to study about these things. But in India, Dr. Pradeep K. Divakar was initiated all the case. And here he is the molecular phylogenetic sufficient with trentifolia, one of the minute algal component which will be present in the diorygma. Only in most of the crustose lichens will be found with the trentifolia. At the same time, here the 24 diorygma alum will be studied about by Mr. Ansel. Now he is the currently he is the researcher in Agartha Research Institute. And in our case, this Diphlolabia Avzeli was the research was done by Robert. Sorry, not Robert Looking. He is like Indian Robert Looking, like that. Rajesh Kumar, KC Rajesh Kumar have been done. He was supporting and taking the advancement of the graphidase, not only the graphidase, including because already the formulase have been almost done by but in India, still though so far the myth is going on, but those two people have been broken about these things. This remoter trichina is nothing but is a Parmelia lichens only. But here also we have been found, and these are all my research about the lichenoids. Lichenoids means nothing but the fungal component cannot be produced, not only the fungal component, including the algal components. While we are studying about the lichens, the lichen secondary metabolites have been isolated from heteroderma diademata, leucodermia leucomelas, and Permotrima aurata, and Permotrima reticulatum, and Stichta fuliginacea. So these five lichen species have been taken, and we have found more than 80 metabolites individually. But in this case, everything will be tested with the cancer cell line, but only few things like hexa decanol 1,4-methyl and hexagonic acid and propionic acid have been give with a good result. And the artificial intelligence has been tested with my research during my doctoral studies with the protein receptor and the lichenoids, which is called secondary metabolites of the lichens, benzoxycyclohexidazine as well as the acetamide from the Permotrima reticulatum as well as Ramallian achondroplichens. It has been going on that. And it was tested for anti-diabetic activity with minimal concentration and with the cell line of R1 and 5F. We called it as RIN 5F. So in this case, the cell was regenerated with the support of the lichen metabolites, and ultimately with this kind of materials, particularly the 2-methoxybenzyl alcohol and 6-epi siphonol and guanosine, and these chemical components molecular weight was so less, below 500. But in that case, we have been curious to study about this because of the molecular weight was very, very less, but their activity was very high comparatively to any other living or non-living materials metabolites. So ultimately, I'm going to conclude this because I think it was very fast things. So Department of Science and Technology, behalf of the Indian government, they have appreciated about the studies because the ancient symbiosis was found from the Western Ghats, which means globally the Earth was connected with not only by the network, including the underground network like mycelium. So this research have been conducted with the guidance of Dr. Rajesh Kumar KC, and ultimately, so far, few of the things in volcanic area also have been found, but during the short period, I cannot conclude this. I'm sorry. And in future, and so far, there are some researchers have been found with oceanic area, particularly the Indian Ocean and the Deccan Bay of Bengal region. So we have start to study about that because Dr. Sanjeev Nayaka and their team have been tried to hunt the lichen with my professor, the doctoral professor, Dr. P. Ponmurugan. They have tried to hunt some amount of lichen in particularly the Rameswaram and Pamban bridge area, but they couldn't. But after that, they have collected few of the uh, um, Particulus lichens, which was growing on the surface of the tree trunks, particularly the neem tree, Azadirachta indica. They have found a few of the graphics, and it was studied by Dr. Siljo Joseph. And all the things have been happened with the support of Dr. D.K. Upliti, so former director. And so in future, the lichen materials, and recently I have curious with Dr. Belishinai, and he was working on oceanic, or the marine fungi. So possibly we have the hope to study about the marine lichens also. So this can be happen with the Mariana Trench, one of the deepest place. So it already going on. So in this case, we would like to continue the research with future generations. Yeah, thank you. So can you please ask
Yeah,
anything as you want?
thank you, Dr. Rajaprabu. Can you please stop sharing so that we can see you and we can see you on video also? Can you please switch on the video, Dr. Rajaprabu?
Yes, sir.
Thank you. That was very insightful presentation. I like the slides. I like the way you explained the things, and I finally ended with the an ocean of opportunity. But the thing is, lichens need the cyanobacterial or algal partners, right?
Yes, sir.
And
Yes, sir.
and I
Definitely.
just saw that you talked about Mariana Trench. I mean, I understand you didn't mean that you could find lichens there, but at least some of these lichen species can withstand pressure as high as 1,000 atmosphere. That's what you meant. As I remember from my conversation with Dr. Sanjeev Naikam, these marine lichens are restricted to the intertidal regions, or at least the coastal regions.
Yes, sir.
Have you thought about deep marine lichens in deep sea? I mean, they are photosynthetic, right? The algal part or the cyanobacterial part.
Yes, sir. Actually, in my view, most of the
Let me
cyanobacteria can be found
let me
from.
continue. Because if you go below 100 meters, there is no sunlight. The sunlight is different. Don't find energy. What you
But
call sunlight.
possibly nitrogen content can be helpful to grow on the deep ocean, sir. So that is what.
You mean chemosynthetic activity?
Yes, particularly because it is not only the temperature tolerant, is always saline tolerant also. So sometimes it is like air bubbles, air bags of some of the macro algae. So possibly this can be grow inside the ocean, or else some of them can be living as a bacterial components. So in that case, lichenized bacteria as well as the lichenized fungal components can be sufficiently have the ability to survive. But it is like in a sleeping mode because many of them in the high altitude region, many of the lichens are only in the sleep mode. So during winter and the rainy season, monsoon season only, they are get ready to survive, and suddenly it start to grow very vigorously. So all the research have been in the field based. I have observed. So that is why I have the thought, but it may hope, but ultimately how to prove it. So these are the things to be in future. It has to be done, sir.
See, Dr. Rajaprabhu, you are an expert in lichenology, and other than the curiosity part of it, just can I have two reasons why should we spend money on marine lichens? Actually? Because I am from National Institute of Oceanography. Tomorrow,
Yes,
if I
sir.
want to initiate research on marine lichens, I would have to convince my bosses why.
Yes.
So, can you help me with two reasons why we should be doing starting marine lichen studies? Actually.
Yes, sir. Particularly the metabolites based, there are so far there are different mm, uh, uncurable diseases are surviving, and it was spread out from today itself. Today morning, I have found about former President Jawaharlal Nehru. Because of him, a disease was spread out to the from Italy to here because of misconducting of physical behavior. So I'm sorry to say this, but whether it is true or not, I think so. But in this case, after the from the museum, they have taken all the things of our former president Jawaharlal Nehru. In this case, we have maximum possibility to cure about the cancer cell lines. Not only this cancer cell lines, because that is one of the most dangerous disease with will be we people are surviving with that. But in this case, not only in the medicinal purpose, at the same time in the agriculture based area. So we have recently China have been found saline tolerant rice variety, but already we have in particularly in the region of Palayam Kotai, which is located in Tinaveli. So they have already living and they are growing the cultivation of rice. So at the same time, in our case, in the particularly the myco, it is like bacteria. Not only the it is a symbiotic sir. So the symbiotic activity, the lichen metabolites maximum found from the mycelial content of the lichen. So the salinity strength can be more efficient than normal fungus. It means individual fungus, but this microbial biota can be produced more not synthetic way in the natural way it was producing, and so far there are numerous evidences are there comparatively unaccounted or else unidentified lichen metabolites which cannot be produced by the other organism in any case. So that is why we can be go for the next generation. At the same time, here the polycated pathway can be playing a major role. Here the CRISPR 7 gene editing research can be helpful for that. So that is why I am very curious about this. At the same time, strong way I have to suggest to carry about the lichen search or lichen contained brain area. So particularly because already in Chola dynasty, Tarangam body, almost Dr. Lokesh A. Raja, he have hunted around more than 40. It was new to India, which means new to India means it was survived in or else identified from South Africa and Australian regions. But suddenly in 2013, he founded and it was appreciated by Dr. Upriti, so Professor Upriti. So that is why where we left, there may be some hidden gems will be there. So that is why I'm having the curious in this case. So I would like to suggest in this case. That's it.
Thank you. Thank you very much for that. I would request anybody in the audience, if they
Okay, sir.
have any questions,
Please, sir,
unmute
please.
unmute yourself and ask questions. Anybody in the audience, please. You can. Okay, Milya, please. Yeah.
Hi, thank you for the presentation. It's very interesting. I was curious because I know some symbiotic relationships, there can be a bit of flux in between the different functions that they have. So I was wondering if the fungal partner ever also plays a part in decomposition or only relies on the algae partner or cyanobacteria for energy. Does it ever, in certain situations, become the, I guess, actor in that relationship to provide the food?
Yes, of course. That is what I said in the beginning itself. Not only the photosynthetic partner like algae producing this, even the cyanobacteria, including the fungal partner, also producing food because the nutrient to be converted as energy and it will be storing in the mycelial components. So it once it holds, then it will be used for the rest of the period. So only few cases, particularly in the cyanolichens, which means like leptogium in this case, there are few jelly lichens are there. So there, little bit possibly, most of the um, cyanobacteria will be produce more amount of food materials, and it was storage by the converted into energy and the storage by the mycelium of the fungal partner. This is it.
Okay, thank you. Yeah, I missed that earlier. Thanks for explaining.
Yeah, thanks for your question.
Thank you. Any more questions?
May I have question, please?
Dr. Munazo, please. Dr. Kiran, Dr. Munazo Kiran.
Hello. Yes, can you hear me? Hello.
Yes, please. You can go ahead.
Okay, I just want to ask in case of corticolous lichens or the lichens which are present in the form of community on the barks of the trees. And in case of lichens, we are familiar that the photobiont is the algal partner. But is there any possibility that some of the nutrients they may be absorbed through the rhizome from the host green plant or like the tree? Is there any
Yes,
study
definitely.
available?
Yeah. It is like an orthodox. So we found in Bharti University. Only few of the lichen species, such as generic level, pyxin and diorygma, sorry, not diorygma, pyxin and drinaria. These two lichens are very common thing in the unpolluted or else the um, publicly easily available these two genera, which will be producing the metabolites. It will be destroying the host as well. At the same time, their rhizines will be absorbing the moisture content as well as the nutrient content from the host, such as Carticolous lichens, not only the Carticolous lichens, including the Lichenicolous lichens. In the Lichenicolous lichen, one of the major lichen, like folios lichens, can be giving some crustose as a habitat host. So these crustose lichens will be growing on the Lichenicolous fungi. So these studies have been available with um, University of Rajasthan, Dr. Um, let me know. Just his name. I just forget his name. Sorry, but he have
Okay.
done regarding
Okay,
that
I
kind
got,
of things. And
yeah.
yeah, please.
Yes, I got your answer. So it means that um, means in general we read in text that it's a bipart relationship between the algal and the
Yeah.
fungal partner only. But as you have suggested many examples, so there is a possibility that it's not
Yeah. Sure,
rather
sure.
maybe it's not a balanced symbiotic relationship. It may be weak parasitism or something like that. There is some possibility, something like that.
Yeah. Sure.
Thank you, sir. Thank you.
Yeah. Welcome.
Any more questions, ladies and gentlemen?
Sir, good afternoon. I have one question. Yeah, Sir Rajababu
Yes, ma'am.
sir, your
Please.
presentation is very nice. So we got some
Thank you.
different points from lichens. So, sir, as you said, actually, apart from algal and fungi, some bacterial cultures were also detected in your sample. So, which you have
Yes, ma'am.
identified in at different places. So, my doubt is that, so is there any symbiotic relationship between bacteria to the fungi and algae, or
Yeah.
what is the benefit of that bacteria when it lives with the algae and fungi? That is my doubt.
Actually, in our study, the bacteria need some host because most of the bacteria will be the airborne bacteria, which will be surviving in the air generally. So once it will be spread out or else migrate from one place to another place. So it was searching some host, and once it attached, their genetical material will be passed through their connection. So particularly once the algal lichen formation can be happen particularly first profoundly the algal component will be attached in a particular host, and the mycelial component also can be spread out because it is a internet connecting way. So once this will be connected, then automatically by chance only, the bacterial components can be attached with this, and it producing, and it has some ability like tolerant ability, like stress, not only the stress tolerant, including the thermal tolerant, particularly ultraviolet tolerant has been found with many of the recent studies with bacterial strains. So in our case, particularly the actinomycetes bacteria, which means yes, actinomycetes. Yes, the particular actinomycetes have been used as a probiotic bacteria in different case, but in this case, we have isolated some bacteria in endolychenic formation. So once we started to culture the lichen separation symbiotic association, so we found the bacterial strains also, but it is not man-made error. So I can surely say, and it gives some stability to control their temperature, the room temperature it was surviving, and it gives the nutrients to us, to the lichenized fungi also. So this is what happened. And one more suggestion to you to be studied in the case. So there are many cyanobacteria is there, which means the colorful bacteria. In during 2018 to 19, before pandemic situation, I found the pink color bacteria and purple color bacteria, which will be found in the lichen. So it can be studied, or else it should be studied. It will be appreciated to the research community. Yes.
Thank you very much, sir. Thank you so much. Shana, sir, thank you. Thank you for
One last question from the audience, or Nilam, please. Dr. Nilam.
Okay. Can you hear me?
Yes, please.
Hello. Oh, yes. Thank you. The presentation is very interesting. I have 4 questions.
Yeah, please.
First, how can lichen be cultured and applied for anti-diabetic purposes, considering that lichen are a symbiotic association between fungi and algae, as you say also have bacteria also. So anti-diabetic investigation use the whole lichen fruiting body or each partner?
Yeah, but we have found the secondary metabolite by elution of different solvent methods like acetone, chloroform, and petroleum ether. These materials have been used to isolate and particularly purify the compounds. There are many different secondary metabolites have been found, studied by Ahmad Jian and Orange and White. They have giving them, particularly the Culberson, have been formulated the extraction method in 1972, and from the usage of a different solvent for the you can refer also, no issue. And in the secondary metabolite isolation, we have pure compound like acinic acid and lacnoric acid and cechic acid. These are all the very common secondary metabolites which will be used for different antimicrobial and biomedical application, including the biotechnological application. So we have tried with that. So that is why after concluding the minimum inhibitory concentration, we have tried with these things. So earlier it was tried with the antibacterial activity, then only the antioxidant activity has been conducted, then the anti-diabetic activity have been conducted in silico as well as the in vitro method. So ultimately we found this. So these secondary metabolite can be useful once in the symbiotic association only it can be produced. If it is separated, the endolychenic fungi is giving the different metabolites as well as the algal component also giving in a different metabolites. So that is why. So there are many possibilities to extraction as well as the purification, and ultimately it gives them pure compounds, gives the curious to study, not only curious, including the once it was completely study about these things, we can go for the further patent itself. That's
Dr.
So,
Nilam,
which component of the lichen is more suitable for anti-diabetic application? Which
yeah.
component?
Which
Fungal
component
partner
particularly?
or algae photobiont partner
This
or
is what I
associated
said.
bacteria? Which
In
one?
associated by
Okay.
actually in this case, different in my study, the endolychenic fungi component will be giving the good result. Remaining particularly the compound techic acid of Ramalina conduplicans, it gives a good result. So you can, it is in the symbiotic association only.
My
Dr.
last
Neil?
question, please.
Yes, please. Yeah.
Can? Yes. Okay. Thank you. If anti-diabetic screening require large quantity of lichen fertility, for example, and what your strategies can be used to obtain sufficient biomass? We need more, like, for example, for to enhance the biomass. Can
Yes, you are right.
lichen be
That is
processed
right.
so that some material can be used repeatedly for extraction and purposes other application? Thanks.
Sure. Actually, your question is, we need to conserve the lichen as well as the biodiversity should not be destroyed, but we have to cure the disease as well. So in this case, what we have to do, bioreactor is a very good technology to once the isolated compound, particularly in the lichenized algae, it means algae or else the endolichenic fungi, we can be cultivate in the bioreactor, then we can go for the metabolite extraction. There are many advanced technologies are there. So we can use in this at the same time, once you know the particular compound, we can go for the organic synthesis of the
Oh, no.
secondary metabolite.
Okay, thank you.
Yeah, you are welcome.
Thank you. One last question from Sana. Sana, are you there?
Yes, sir. Actually, I want to ask, can we use lichens in deforestation, provided the rate of growth is very slow?
Yeah, it is already the research was going in Brazil and France.
Okay, sir. Thank you.
Yeah, welcome.
Thank you, Dr. Rajaprabu. We will
Thank
have
you,
more
sir. Thanks for
discussion.
your wonderful opportunity. You gave me the
And
very
we
good
have
thing.
experts on the editorial board of Micronesia. We don't get many manuscripts on lichens, but whenever we get good comments. But we would like to strengthen that section of lichenology on that editorial board. I hope you could help us with this in future.
Thank you, sir. Definitely with your support.
Thank you. And ladies and gentlemen, thank you very much for your kind participation. As I put it, I have already put it in the message box. If you want a participation certificate, please submit this Google form. And ladies and gentlemen, thank you once again. Have a good Sunday wherever you are. And see you soon, probably next Sunday for the top 3 and 4 of Micro Asia and Web Seminars 2025. Bye-bye.
Yes, sir. Thank you. Thank you so much. Wish you all the best to all.
Thank you, sir.
Thank you.
Thank you.
Thank you, both the authors.
Thank you. Good afternoon.
Thank you, sir. Thank you.
Thank you, sir.